All right, chapter 10. Chapter 10 begins going along with that same idea that smaller is better, but I want to go into a little more detail about it. Plus, it gives you a chance to talk about a little math, which I know you guys all love. Okay, I got 3 boxes up here. I'm not even using units. It could be centimeters, it could be whatever, but I don't even care about that. I have 3 boxes up here. Small, medium, and a large box. And if I want to determine the surface area of box number 1, I can do that. Okay, and I won't go into the details of how you do that, but if I took the surface area of box number 1, it would come out to be 6. What is surface area? Surface area is the amount of area on something that's actually exposed. It's on the surface area, okay? So surface area for that 1 box is 6. This is like a cube. It's like a 6-sided cube. It's 6. Box number 2. Box number 2, it's bigger. So it makes sense it's going to have more surface area, 24. Box 3, even bigger. Surface area, huge. In other words, the bigger you are, the more surface area you have. Just even like this ball idea, the big ball here compared to the little ball, this big ball has more surface area. Makes sense. It has more area that's actually exposed to the surface versus this. But Mrs. Hatcher just said that this is the better cell, and it is. Because you not only have to take into consideration the surface area of something, you also have to take into consideration the volume. So, what I've done here is I've determined the volume for each of these 3 boxes. Volume number 1 is 1. Volume number 2 is 8. Volume number 3 is 27. In other words, volume. Volume is how much, space it takes up. It makes sense that the bigger box is going to have a bigger volume. It takes up more space. Just like in this ball right here, this has a bigger volume than this. It takes up more space. Okay, but when it comes to the most efficient cell, here's what the deal is. Now, this gets a little tricky. Bear with me. Hold on, let me erase this and kind of go over it with you. In order to determine the best box, you have to be able to understand what a surface area to volume ratio is. Okay, this word ratio is just a fancy word for a comparison. Surface area to volume ratio. Okay, so I'm going to compare the surface area and the volume to each other, is what that's saying. Well, for box number 1, I had a surface area of 6. I had a volume of 1. So I write it like that, 6:1. That basically means I'm comparing those 2 numbers. Surface area to volume ratio over here, I'm comparing these 2 numbers, 24 and 8. However, I can reduce that. When I reduce that, it becomes 3:1. I can't reduce 6:1. That's why that one says is. 6:1, 3:1. Box over here, 54 to 27. Can anybody reduce that for me real quick? It's real easy. How many times does 27 go into 54?
Twice.
Twice, 2:1. All I did was take that number and reduce it. Okay, so I've got these 3 ratios, these 3 comparisons. And when it comes down to which cell works the best, cells want a high surface area to volume ratio. What that means is you want to have a lot of surface area, but not a whole lot of volume. You want a big surface area number and a little volume number. And that's exactly what you have in this cell number 1 right here with 6:1. 6, a very high number, 1, a very low number. Cell number 1 wins. That cell is going to be the most efficient at getting substances in and out of them. Well, let's think about it this way, guys. If you have a cell Remember what we learned last chapter. All cells need to make ATP. And a large way that's done is a process called cellular respiration. And when a cell undergoes cellular respiration, they're going to make a lot of CO2 waste product. Well, cells do not like having carbon dioxide inside of them. It's toxic in high quantities. You better find a way to get that CO2 out of there. Well, if the cell is super big and has a large volume, that's going to be hard to do. But a really small cell is going to do a better job of doing that. So that's what I mean. Smaller the cell, it's going to be able to get that CO2 out of there as quickly as possible. So cells want a high surface area to volume ratio. They want a lot of surface area, not a lot of volume. So what does that have to do with this chapter? Well, that's why cells divide. You know, when a cell becomes kind of big, it says, all right, if I want to be more efficient, it's going to make a lot of sense for me to divide into two. That's good. That's good for a cell. All right, so, cell division. That's the whole name of the chapter. Cell division. Process by which a parent cell divides into two or more daughter cells. All right, so a little bit of language here is different from what we're used to. I talk about a parent cell and I talk about a daughter cell, but it has nothing to do with male or female. They just use these terms kind of generically. A parent cell is a cell that's going to decide it needs to divide. So a parent cell is like the parent. When it divides, the cells that it divides into are called daughter cells. Now those daughter cells are going to go on and grow and become useful and do everything. And eventually those daughter cells are going to become parent cells, but that's down the line. Okay. Process by which a parent cell divides into two or more daughter cells. Oops, I should put an S on the end of there.
Process by which.
Oh, thank you. Process by which a parent cell divides into two or more daughter cells. Thank you.
You notice that you make all your A's capital?
Yes.
No, it's usually every other one.
I've been called on that before, but every once in a while, like in the word parent, it's lowercase.
It's every other one, I think.
It's weird. I don't understand it.
It is.
I know.
It's parent cell and then daughter cell.
Yeah.
And then she has parent.
And I am so oblivious to it, I don't even know I'm doing it. So, kind of funny.
Chucky makes all of his letters like capitalized.
Does he?
But he'll make them like lowercase capital, so they're smaller than actual big. I don't know. I don't know.
All right, you guys should really know the difference between these two words. They refer to types of cells. A prokaryote is a very simple cell like a bacteria. The DNA is not in a nucleus, so there's no nucleus. And a eukaryote has a nucleus. Very good. Okay, so but now we're talking about cell division. So we're past the point of what they have and what they don't have. But the reason I bring it up is the prokaryote, like a bacteria. Again, that would be an example of a prokaryote. They're very simple. When they decide that they need to divide, okay, and they do. Bacteria definitely divide. There's one huge difference between prokaryotes and eukaryotes when it comes to their chromosomes and DNA. The chromosome of a prokaryote is circular. I'm going to say it's a circular chromosome, and I kind of drew it in here. It really is like, there's no nucleus, but the chromosome itself is very circular. In a eukaryotic cell, it's different. So all the cells in our body, number one, there's a nucleus, and the DNA that's inside of our nucleus is not arranged in a circle. It's actually, they're linear. So they look like little lines. So I'm going to call this just linear chromosomes. Okay, now this is a big deal. Now, obviously, this is not a human. I have a cell here with 4 chromosomes. That's what these 4 little lines are. They're linear. They're arranged in a line, in other words. It's not human. Humans have 46, so obviously this is not a human chromosome or human cell. But I didn't want to draw 46 little slashes there. Okay. Here's what the deal is. Prokaryotes, because of their chromosome partly, they divide a very different way from us. They undergo a process called binary fission. Fission. We should remember that fission word from last year. When we were talking about atoms splitting up, it was referred to as fission. Remember, we talked about fission, then we talked about fusion. Fission is the atoms were kind of breaking apart into smaller atoms. Fusion, the atoms were combining to make bigger atoms. This is kind of, I guess you can kind of look at this word the same way. Binary fission is basically where a prokaryotic cell is literally going to take that chromosome, make a copy of it, and stick a copy of it into 2 cells. In other words, the cell's just going to split, become smaller. In other words, this chapter does not focus very much on prokaryotic cell division. They tell you that its name is binary fission, and that's important, but it's really quick, it's really fast, there's not much to it. It's a circular chromosome, easy done. But this is one of the reasons why bacteria reproduce so quickly, like they do in hospitals, like they do for infections. Part of the reasons that bacteria is such a problem is because the way it reproduces is so quick. Okay, so that's kind of a problem. Eukaryotic cells like animals and plants and fungus and protists and all these other types of cells that are familiar to us, the chromosomes are arranged linearly. So basically, that's going to become more of an issue. It's not as easy as what you see here on the left. That's why when a cell has these kind of chromosomes, it has to go through a different process that we'll talk about in this chapter called mitosis. Okay. I want to kind of focus a little more on those chromosomes for eukaryotes, eukaryotic cells. All right, I'm going to draw 3 cells here.
Should we write that right underneath our...
Yeah, I would write it right. It just kind of goes along with the eukaryotes on that right-hand side, but I just needed more room. I'm going to draw 3 different eukaryotic cells. Obviously, I drew a cell and I drew a nucleus in all 3.
They look the same.
Yep, that's what I'm going for, except I kind of ran out of room, so one looks kind of squished, but that's okay. Okay, they're supposed to be, that's okay. Cells don't all look perfect 3D shape anyway, so. All right. This is the funny thing. I want to talk about the word chromosome. Kind of leave some space and write the word chromosome. Your DNA that's inside of you isn't really all that pure. Your DNA is actually kind of, it looks like a twisted ladder, okay, which we'll talk about in the future, but it's also surrounded by a bunch of protein. In other words, your genetic material is kind of gloopy. It's a bunch of DNA and it's a bunch of proteins that are stuck to it. It's kind of goopy and gluey. Your genetic material, like chromosomes, it really takes on very different forms depending on what stage in the cell cycle you're at. Here's what I mean by that. Sometimes in your cells, and this is hard for me to draw with this kind of marker, your DNA, you can't even see it. It's very thread-like.
Is that just supposed to be thin lines?
Huh?
They're just supposed to be thin, right?
Yeah, all I tried to draw basically your DNA. That's it. This is a nucleus, by the way. And the DNA inside of it is hardly visible. I'm going to call it thread-like, invisible genetic material. If I would pluck out a cell from your guys' body and actually look at it under a microscope, there's a really good chance that that's what your genetic material would look like. It would just be real thread-like and hardly even visible. However, as a cell, and I'm going to draw an arrow from left to right, gets closer to cell division, like in other words, once that cell decides that it's time to divide and make more of itself, the chromosomes change form. In the second picture, I'm going to draw the chromosomes just like I did before, kind of looking like these little tiny short strands. Those are, I'm going to refer to those as condensed chromosomes. They're chromosomes because now this material that you saw in the far left picture, it's the same material that you see in this middle picture, but all those chromosomes did was shorten up. They kind of crunched together. When they kind of crunch up together, they become chromosomes and they get really short and fat. Okay. Good. So basically the same material that's here is here, just in a short form. However, we're going to see when we go into the details of mitosis and meiosis, which we will soon, you're going to notice that that genetic material takes on yet another shape. Those 4 chromosomes are actually going to make copies of themselves, and it's going to glue that copy right up to it. In other words, this picture on the far right is showing, I'm running out of room here, duplicated chromosomes. In other words, I took the chromosomes from the second picture, like a line, I made a total copy of it, and I stuck that copy right onto it. And that's why you see that each of these chromosomes over here has like like 2 sides to it. It's like an X. A lot of times they show it looking like an X. I still only have 4 chromosomes in this third picture. I have 4 chromosomes in this second picture. I have 4 chromosomes in this third picture. The third picture just has that same chromosome kind of copy, but they still consider it 1 chromosome. 1, 2, 3, 4. That's hard. I realize this is kind of difficult stuff, guys, but this is kind of important to understand when we go into the details. All right. Again. Good. Any questions on that so far? So, in other words, your chromosomes, your genetic material, your DNA looks very different depending on what stage it's at in its life cycle. Speaking of life cycle, this next picture I'm about to draw, I need your brain for, big time.
Oh, gosh.
Because this is probably going to be one of the most important pictures I draw all year long. Okay? This is important not for you just to draw it, to really understand what I'm drawing next. Because this is going to kind of set the stage for what I'm going to talk about in the future. Here's what I'm going to title it, the cell cycle. Okay. This is going to basically be referring to, for a given cell, how does it spend its life? What does it start off doing? What happens next? What happens next? I'm going to represent a cell cycle. I always say this every year. Pretend that's a pizza.
Oh, now you're making me hungry.
I know. I've got a big pizza here. And I'm going to divide that pizza into 3 big, giant slices and 1 tiny slice. I'm going to name the. We got to start somewhere on this life cycle. So I'm going to start right over here. I'm going to say this arrow, with a little star next to it, represents that point in time where a daughter cell has just been formed. A daughter cell just formed from a parent cell. Bam, it's a new cell. Well, that cell is going to spend the first part of its life in something called a G1 phase.
That little star's a daughter cell.
Yeah, that little cell is just the point in time where you got, here, I'll even write it. You have a new daughter cell. And that new daughter cell is going to spend the first part of its life cycle in a G1 phase. And the only thing that daughter cell is going to do in the G1 phase is it's going to start to grow. But look, that's a pretty big piece of pie. In other words, that cell is going to spend kind of a long time doing that. Okay. After it gets done in the G1 phase, it's going to head over to the something called the S phase, and something really important happens here in the S phase. All of the DNA that's inside that cell is going to duplicate itself. It's going to make a copy. I'll even put that in parentheses. It's going to copy itself. In other words, all that DNA, it's like going to go over down the hall to a copy machine, make a copy of it, and just kind of store it there for a time being. Okay, well, I kind of just drew a picture. Hold on. I'm going to go back over here. This picture I just drew. I just told you in this last slide that sometimes chromosomes look like this, and then sometimes they actually make a copy of themselves. They go from that to that. Well, guess what, guys? That's exactly what happens in the S phase. The chromosomes are going to go from that form to that form. They're going to change forms in that phase. By the way, I'm not even close to mitosis yet. This has nothing to do with mitosis. I'm kind of laying the groundwork for mitosis to take place, but this is not mitosis. All right, the cell is going to continue its track and go into something called a G2 phase, and the only thing I'm going to say about that is, all right, now this cell is going to start to prepare to divide. This cell is now, okay, all right, I got to divide soon. This little tiny slice of a pizza, I'm actually going to divide again. I'm going to make 2 slices out of the tiny slice. The first slice, the one that's bigger, is going to be something called the M phase. All right, now we're getting somewhere. The M phase, I'm going to write it down here because I don't have enough room. The M phase is also called mitosis. So, Mrs. Hatcher, what you're telling me is mitosis is just a little tiny slice of pizza in the big scheme of things? Yes. In other words, a cell spends very, very little time in mitosis. But that's funny because the chapter in your book tends to spend the whole time talking about mitosis, but when it comes down to it, a cell just really doesn't spend that much time in it. By the way, mitosis, what it is, this might also catch you off guard. What the technical definition of mitosis is just the division of the genetic material. In other words, your DNA divides up in mitosis. The M phase we will talk about in greater detail a little bit, but mitosis consists of 4 parts in this order. The first stage is called prophase, the second stage is called metaphase, the third phase is called anaphase, and the fourth stage is called telophase. We've got to remember those words. We've got to remember them in that order. We have to remember that those are the phases involved in mitosis. We will go into detail about each of those phases another day. For now, just understand that you have to understand these. There's an easy way to remember these. We'll get to that later. Yeah, PMAT, PMAT. You got to remember that. PMAT. You got to remember that. I don't care if that's how you remember it. PMAT. PMAT. Okay. There's a tiny sliver over here. Ooh, I'm not going to be able to write this word in. CYTOKINESIS. That teeny tiny sliver is something called cytokinesis. Let me write the word again down here if you didn't get that. Cytokinesis is division of the cytoplasm. You guys remember what cytoplasm is? It's kind of the goo that's inside the cell. Here's the kicker. This is important. Mitosis is going to result in a division of the genetic material. But when you're done with mitosis, like at the end of telophase, you do not have two new cells. Oh, wait a minute, Mrs. Hutcher. I thought mitosis is just division of the DNA. You haven't created two new cells yet. You still have one cell, but it has the same genetic information on two sides of the cell. It's not until cytokinesis where that one cell is actually going to go bloop and become two cells. Cytokinesis is where you actually have division and you're making new cells. The whole process of mitosis is just there to divide up the DNA and the genetic material. It's very, very different. Okay, one more thing. We need to one more big word, and then I'm approaching the end here. All right. You know how some pizzas kind of come with a crust of cheese, like cheese crust pizza kind of deal? Well, I'm making a crust pizza over most of this, but the little sliver right there. And I'm going to give that cheese crust a name called interphase. Ooh, it has that phase word on the end, but please do not associate this with mitosis. It's different. This makes for a great test question. But before I do that, let me tell you what it is. Interphase refers to the time in a cell's life when it is not dividing. It is only during this portion right here, which is mitosis and cytokinesis, where you are actually have cell division going on. All the other time a cell spends in its life is in interphase. And that just refers to the time in a cell's life when it's not dividing. If I took a cell out of your guys' body right now, chances are it's not dividing. It's just sitting there doing its thing that cells do. Making proteins, making carbohydrates, making some lipids, keeping you happy. But it's not dividing. It's actually only dividing in the last little kind of portion of its life, in M phase is cytokinesis. Okay, so that's actually really important. I actually want to add something up here. A cell actually spends 90% of its time in interphase. In other words, cells only divide for a very small part of their life. Okay, there's a lot of big words here, but this is actually a really important picture. Let me actually show you some pictures. We're done writing. Let me show you some pictures covering the information we talked about today. By the way, that same picture I drew is important enough they need. You good? Yep. This picture is in your book even too, and again, it's the same thing I drew, but I can't stress enough how important this picture is. A great test question would be, what phase does a cell spend most of its time in? You would say interphase. Another good question would be, the process of cell division is a two-part deal. Name those two parts. And you would say mitosis and cytokinesis. Okay. One more picture. This picture is kind of nice. Let me just show you one at a time here. Don't even look at the bottom part yet. Let's look at the simple picture first. The whole point of this slide is to compare prokaryotic and eukaryotic chromosomes. In other words, right here you have a picture of a prokaryotic chromosome, that blue thing. It is just one piece of big circular chromosome, and that's it. However, eukaryotic chromosomes are much more complicated. They basically are packaged as these units called chromosomes. And they show you what this kind of final chromosome looks like over here. This picture also goes on to show you what makes up a chromosome. They want you to understand that it's not just the DNA. This colored thing is DNA. This twisted ladder is DNA. But they want you to understand that the DNA is actually wrapped around a bunch of proteins. And together, the two of those things make up a chromosome. Do you guys know how many chromosomes humans have in each of their cells?
36.
46.
Oh, I had a
Okay,
six.
46. In other words, what that means is if I would take any cell out of your body, whether it's a liver cell, a skin cell, a cardiac cell, a muscle cell, doesn't matter. If I took any cell out of your body, every one of those cells is going to have a nucleus because you guys are eukaryotic. And inside that nucleus, you're going to have a bunch of chromosomes. You're going to have 46 of them. So you have 46 chromosomes in every cell in every cell in your body. Okay. And they kind of all tend to have this general shape.
My calls for an earthquake.
No, I'm not doing that.
All right. I think that's all for today.
Do it, big boy. Earthquake. Do it. I didn't do it.